colleague, Erdem Özkaya, who studied first his bachelor's degree at Istanbul Technical University, and then progressed to Ludwig Maximilianus University of Munich, and he met with genome engineering for the first time. Then he came to Graz, which is really nice, and he's now doing a PhD at the Hematology department in the Medical University of Graz at Andras Reinsch's lab. So give a warm welcome to Erdem.
I will also try to really talk loud. Hopefully it's fine for that. Yeah?
It's fine because you're looking at me.
No, I was looking at him, yeah. So thanks everyone for coming, and thanks for the PintofScience team to arrange this. I think it's a really cool idea to talk about science and give information and see how it can be shared and people will have an idea. So my presentation is going to be somewhat different than Professor Thomas's presentation because I didn't know who will come. So I started from scratch, like how many cells we have, what are the DNA levels, how are they doing. So it's really more of a lecture kind of thing. Hopefully it's going to be like, it's like high school information. Some of them, hopefully I will do better than my high school teacher to really give you interaction. And the one thing is that I have a couple of questions inside that are interactive. So when you have your phone, you can actually scan the QR codes and you can enroll. So I'm just going to give it a bit time so you can come in and do live answering. So all of the answers will be happening on the screen. So I have 5 questions and hopefully that will be good. I tend to speak fast when I'm nervous, so if I'm going fast, tell me something. I'll go down slowly. And yeah, so let's start. So with the overview of the presentation, I'm going to first talk about the body, cell, DNA, and then I'm going to talk about cancer, treatment problems, and AML, and then genetic engineering. How are we using it? What is it, first of all? Because we all hear it, but most of the people doesn't have any idea how it's working or how we apply it to the science. First, I'm going to start with the body, cell, and DNA. So I'm going to start with a fun fact. Adult human body has approximately 35 trillion cells. This is 35 followed by 12 zeros. So it is the amount that is like super crazy big. Like human mind cannot even comprehend. It's just so many cells. You say, yeah, there's a lot of cells. But if you think about it, it's a lot of cells. And first, I'm going to start with the first question. Actually, that's a bit of a problem that I need to come here every time. But you can scan the QR code. And then you can answer the question. So I just have like a ranking question, like what is the most common cell types in our body? Is it like the skin cells? Are those muscle cells, brain or neural cells, blood cells, skeletal cells, fat cells? Just to give an idea to see how it is going to be. So it's going to be pretty interactive, and you should be able to see how many people join. And if it does not work, please let me know because it's...
How do we rank it from most to least?
Exactly. So whatever the most one was the first most common one, and then go down to the low. So we'll see what are people thinking, what we have in general. And after this, you don't need to reload again. You can stay on the page. And when I go to the next question, it should appear in the same page of the mentimeter. So it should just stay in there. If not, you can scan the QR code again. And if you reach 20 people, I will continue, just for the sake of time, because I don't know how long it's going to be. Okay, it's getting close. Okay. Yeah. Okay, 20 people is good, I guess. Okay, I'm just going to continue with the question. It's actually correct. The most common cell types are blood cells. Because we don't think about it, but we have skin cells that cover all of our body. We have muscle cells that are basically having most of our the muscle system. And we're thinking that they're going to be the most common one. But actually, if we... But it is too close. Yeah, okay, now it's good. So, to just give an idea, you don't need to read everything. This is just an overview. On the left side, we have the cell count of like all of the cells that we have. So basically, what you see in here is just the numbers, and you see how many white blood cells we have. It doesn't count including the red blood cells, that's the cells that are actually carrying oxygen, because red blood cells and platelets are actually 4 times more than all the other cells. So there will be 4 times more circles in the outside. But you see, like the epithelial cells, the skin cells are there. The brain cells, the glial and neuronal cells are here. You see adipocytes, the fat cells, and you see small percentage of stem cells that we are going to talk about. On the right side is the biomass, like how heavy they are. And it makes sense because on the back, you see there's another circle. This is our skeletal system, so it's half of our biomass. And also we have a lot of adipocytes. This is like the fat tissue that you have. But you see the red blood cell, these cells are the smallest cells possible. Their only work is just to carry some oxygen, but still they are heavier than your brain, for example, in your body. Just to give you an idea. And next question will be, thank you. It's a quiz, so it's like a bit different. You need to press start quiz. It says maximum points, but yeah, we count the points just to have an idea. So, how many blood cells produced per day? What are the turnover rate? Because we talked about all the cells and blood cells are super common. We have 30 seconds, so we can just give the answers, and at the end, you're just going to give us the answers. So, I started with 10,000, 1 million, 100 million, 1 billion, 100 billion. At some point, the numbers are just doesn't make sense anymore. Numbers. I'm curious though. Perfect. And okay, wow, a lot of millions. And you see the difference between 1 million and 100 billion. The difference is 100,000 times more. It's like it's 100,000 times more than 1 million. Just to give you an idea how big of 100 billion is. This is crazy amount. This is really the amount that the body needs to produce every day. Just imagine that you are having a trillion of cells, and just your blood cells are producing 100 billion. This is the amount is really crazy. I was like getting excited every time that I see it. Yeah. So another fun fact. Since the starting, like the beginning of this presentation, actually, all of your bodies have produced already 500 million cells. Just now, just 5-6 minutes. And this amount is really the crazy that when you think about it, it's just like crazy all the time. You don't think about it, but your body constantly regenerates, constantly turns and making cells, and it's really crazy amount also when you think about it. But how does it do it? How does it actually keep up with this amount of cell production? So I'm going to go with the blood hierarchy. So we have all the different types of blood cells. You don't need to know them, just to show them. Like we have some lymphoid cells, some red blood cells, these are myeloid cells, and they have kind of like, let's say, generals. We have progenitor cells that produce all of these cells. These are not stem cells, these are not mature cells, these are something in between. They can regenerate, they can create all of these cells. That's why they're called progenitors, but of course, on top of all of these, we need to have stem cells. In a way, you can say that stem cells are the queen. It's kind of like the hive of the ant colony, let's say, because most of the ants or the bees, the queen is to produce all of these other organisms. And in this case, we have stem cells that are producing all of the cells that you have in your body. All of these have 500 million cells every 5 minutes, or 100 billion cells every day. But how does it do it? Because when we look at the cell count part again, the stem cells are just like a small percentage of the cells. So they are really small, but they need to make most of the cell count in our body. It's just really a lot of work when you think about it. And it is causing some problems, because I'm going to explain later. If you remember from the high school, to have second cell, to have a daughter cells, they need to go through mitosis. They need to go through cell division. And in cell division, you need to replicate DNA and all of the cell internal parts to have the two cells. And then you have the two daughter cells that are looking, they should be the same. Let's say you have the original DNA, that everything is correct. The problem is sometimes your body is making mistakes. And then you have an error in the replication. And then you have this base pair mismatch. So it is not working out nicely. And the problem is, this mismatch will carry on to the daughter cells. So we have actually mutant daughter cells nearly every time. And this is kind of the problem that I'm going to talk about now. But why DNA or its mutations are so important? Why are we talking about the mutations that we have? You have all the problems in the DNA. You don't need to get UV, you don't need to smoke cigarettes because they can damage your DNA. Why? What is the problem? If you go back all the way to the beginning, this is the central dogma. It's called, it's basically talk about when you have DNA. DNA is basically have all the information that you have. And then you have this temporary information part called RNA, and from these you need to produce protein. If you think about DNA as a book, we can talk about RNA as the page, and then you can think about if it's a cooking book, let's say, protein will be your meal. And this is how your body knows what to do. And proteins are important because this is just a visual information that like our cells are full of proteins. Everything that you have, everything that your body is producing is full of proteins. Whenever you have a problem with one protein, then all of this mechanism might stagger a lot. So it depends what kind of mutations that you have in your body to really keep it up with the proper working of this mechanism. And now, not so fun fact. Mistakes and mutations happen in 1 every 100 million base pair doing DNA replication. It sounds not so much because 1 in every 100 million means that it's just super rare, right? But DNA has 3.2 billion bases, which means every replication you're going to have 32 mutations going to happen. And it means that 32 mutations are going to accumulate. That is the problem. It's not just the happening is the problem because it depends where you're going to have the mutation in, but the problem is that they're accumulating, and I'm going to talk about it later, the AML, the age, is going to become a problem. Thank you. Next question. Okay. That was supposed to go. It's fine. The question is what percentage of our human DNA is actually producing those proteins? Yeah. This is the question that we also remember. I remember wrong, to be honest. When I'm making the presentation, I realized that this is a mistake. And also, I had a friend that was explaining this, talking about this, and then she also thinks that I was thinking something different. So everyone has waters. Let's see. It's 2%. Perfect. Very nice. It is. I guess this is all the bits. I'm not so sure. But still, it was a really good educated guess, I guess. Yes, only 2% of our DNA is actually making protein. And it's just like showing how small the area is to actually make up all the necessary compartments that your cell needed. So your 98% of the DNA will just be empty parts that the mutations are happening in those parts. Mostly, it's not going to cause any problem. But if those happen in 2% parts, then you might have a problem. Yeah. So the second part will be cancer treatment problems, and AML. And another question. This is going to be the workflow question. So why is it hard to cure cancer? What do you think? What are the problems? Why you always say about what are you curing cancer? So we're gonna what are you doing to cure cancer? And now we're gonna see how it looks like. Mutation, yes, as we immortal, nice. Resistance, yes, similar to normal cells, exactly. Expansive, diversity, yeah. Targeting, yes. Every cancer is different. This is a really good point, and we're going to talk about it later. It is really good answer to the parts, and it's basically nearly all of it. Our cancer cells have some mutations, and they become nearly immortal. And the problem is that these are your cells. So your body is actually training your immune cells to not recognize your own cells. The cells that actually recognize your own cells will be destroyed in the thymus or other parts to not make sure that you don't have any autoimmune diseases. And this is making a problem. If you have your own cells causing a problem, how will your body recognize this? This is the main, really hard thing to overcome. Let's say we overcome this. The other part will be that every cancer is different, and I'm going to talk about this. Yes. So the cancer definition, according to the National Cancer Institute, is a disease which some of the body cells grow uncontrollably and spread to other parts of the body. The other things will be cancer cells can start almost anywhere in the human body. Because most of them are dividing. And we have trillions of cells. It can just happen that in one part you have really unlucky mutations that causing the trigger and favorable to the cancer. And the last thing is there are more than 100 types of cancer. The cancer is not just one disease. This is a problem that actually I had it in the normal just bar environment that like one guy came, I said, yeah, I'm working on the research for the cancer. He said, yeah, I heard that there's going to be a vaccine for the cancer this year. Is it true? I said, no, because it's so hard to explain, like, what cancer? Like, what are we talking about? There are some parts that you can actually train the body for it, but the main problem is that it's just so many different subtypes. Even if you have just lung cancer or like blood cancer, it's just so many subtypes that you have. Even in those, there's going to be some additional problems that I'm going to talk about. The one thing will be, same cancer in different patients can be very different. That's why when you have a patient, cancer patient, the drug that generally used for chemotherapy is working for one patient might not work for the other one because it's really different from person to person. And one drug that is happening for the other one will not work for the third one. It's so many subtypes and so many differences. But let's say even if we overcome this, the main problem is even the same cancer in the same patient can be very different. This is called cancer evolution. And this is one of the most common problems that we are having with cancer. Let's say the cancer starts in this one. This is like a timeline, which just goes more on the side. You have different subtype. Your immune cell came and recognized this and killed it. Let's say this is where you're lucky, actually getting rid of like a certain subtype. But then, these cells grow uncontrollably. They're getting more mutations. They're getting more resistance to the immune cells and to the drugs. And they're just like creating this beast inside of you. And that's the problem that like, even if you use a drug, let's say, you get rid of one of the subtypes, let's say this blue parts are gone, you have additional parts. That's why even some people, when they go on the treatment, they have a remission, they are already treated, let's say, and then the cancer relapse. Because we cannot get rid of all of the subtypes. And this is the main problem that we are really encountering, because it just, when it started, it's so hard to really get rid of it. The main thing will be understanding how it is progressing to this area. So I'm going to talk about AML because we are in the hematology group, and AML stands for acute myeloid leukemia. Acute stands for it develops quickly. If you're not getting any treatment in weeks to months, it might be deadly. It is really aggressive disease, depending on what kind of conditions you have. Of course, not all of AMLs are the same. Myeloid stands for the group of cells that are affecting. I'm going to show later, and leukemia stands for cancer of blood. So this hierarchy, you remember, in the right side you have this lymphoid part. It basically stands for your adaptive immune system. When you get like a vaccination, your cells in the lymphoid part actually recognize this vaccine and train your body, and they remember. So in the next infection, you're going to have a training for this. But there's also this innate immune system that you have, or the other parts in the myeloid part. This is the myeloid compartment. In the myeloid compartment, you have already innate immunity to certain kind of pathogens. Like if you have like bacteria, viral infections, and parasites, these cells already know about these, and these are the myeloid parts. And one of the most important parts is that they also have erythrocytes, the small red blood cells that have nearly 4 times more of all of your other blood cells are also in here. And this small part actually, when you have a cut or like a bleeding, these chromocytes are actually helping in covering in those parts. But why is it important now? We talk about this happening, yeah, but how it happens? It happens in your bones. Like when you have a bone, you have this bone marrow inside of you. And this bone marrow is really spongy because these cells need to go into the blood. And the bone marrow is highly circulated system. You have all the blood cells. That all of these produced cells need to go back into the blood. So they need to be suspension. They need to not attach anywhere. But this spongy part helping for this is actually the stem cells are really residing in these parts. They have their own niche, small areas. And from this, you have all the other cells, and then they go into the blood. So you have blood production. What is the problem? In acute myeloid leukemia, the problem is bone marrow produces immature myeloid cells too quickly. Therefore, you don't have actually any more space. It just blocks the production of healthy blood cell development, and so like less space. And basically, if it's causing a problem in the some myeloid parts, you might not have a proper immunity. If it's in the erythrocyte part, for example, that the bloods that are carrying oxygen, you might not get enough oxygen to your organs. And the other thing will be in the thrombocytes. Some AML patients, when they got cut, it does not stop easily, because basically these cannot be produced properly. And this is the problem. This is the part that the AML is so different also by itself, because the AML is just one thing, but it has 4 or 5 different subtypes, and even in those you have different mutations that you need to understand, and just so many, like it's just really getting branchy. The one important thing is that AML, because we talk about all the mutations and everything, the diagnosis, the recognition that you have AML increases with age. So you see in here it starts from 1 and goes to 85, and you see that the incident rate really increases with age. It makes sense when you think about it. You have more cells producing, you have more mutations, so you have actually more chance to actually have this mutation that can cause AML. But one thing that we are actually really focusing on is going to be crazy to maybe tell some information. These are pre-leukemic mutations. They're called CHIP. Name doesn't matter. These are basically mutations that are happened in the healthy cells before actually AML happens. You actually have these mutations, and the problem is just because you have these mutations doesn't mean that you're going to have AML or some other diseases, because the diseases are really varied. You can have hematological malignancy, means like you have blood diseases, you might have cardiovascular diseases, this is also another thing, and reduced lifespan. But just because you have these mutations, again, doesn't mean that you're going to have these. It's the prognosis, like the progression of these diseases are really low. For example, hematological malignancy, just 1% of the rate of all of the cases. And it takes decades. That's the problem. Some people have this and they live and die healthily. It might not be wild to say that maybe a quarter of the people in the group will already have some mutations like this. But doesn't mean that it's going to have any problem in the future. It might be just, you might not develop any phenotype or any problems, and you just live healthily. And this is mostly the case. But sometimes when you have this, you might have a chance to develop additional diseases. And this is what we try to understand. How the hematological malignancy, the blood disorders that we have, namely the AML progression, is happening with the mutations. And the mutation that we're focusing on, TET2, it's just a name. It doesn't matter, but this is one of the mutations that we are trying to understand. Because if we understand how they are causing the cells to develop into the AML, we might stop it before it actually progresses into the AML. And this is one thing that is really, really important, because these mutations, we can actually see them in the sequencing screening of the patients. Sometimes when they come, they're like, what is drawn, and then it's checked what kind of mutations they have. And if they have these kind of mutations, they might be under surveillance, or they might have received some different drugs, because all of them are working differently, and you need to have a drug that is working against these mutations or these problems. Yeah, and the last part is going to be genetic engineering. It's always something that people say, yeah, genetic engineering you're doing, but what is it? What does it mean? Like, how do you do it? What is it starting from? And the first question will be, thank you, what comes to your mind when you hear about genetic engineering?
Knowledgeable child.
CRISPR has nothing else? Wow.
Come on.
No GMOs, no
Therapy.
therapy. Nanotechnology. Adenovirus. Okay, that is quite a good advance. Yes. Okay. Yeah. Seems the case. Well, I guess. I was expecting to have the answer of the tomatoes that live, I don't know, so long in the shells.
But that's true. I think it's where it starts.
Yeah. Fun. Said no one ever. But thank you.
That come from you, right?
So I guess we have really good combination of like answers. It's really well done. It's a really good crowd actually. Because the next thing I'm going to talk about, as you can imagine, is CRISPR. Yeah. So we all talk about CRISPR. We say CRISPR-Cas9, and it's just like if you kind of have some scientific talk or other parts, you talk about this stuff a lot, and you hear this. But what is it? What does it do? Why does it become such an important thing to have? Or like at first, what is CRISPR? If you see in here there's a bacteria, it will be maybe frightening to some of you, or like really nice to know for some of you. Bacteria can also get infection. They get infection from viruses. These are called bacteriophages. These are actually get into the bacteria. They give their DNA, genomic or RNA, whatever they have, genomic information to bacteria to make them produce more of themselves. And the bacteria dies, and then they go infect the bacteria. So they kind of have what comes around goes around. So they have also some infection, but they're not helpless. They have certain mechanism that works against this DNA that they have. This CRISPR comes into play in this one. So how does it work? CRISPR actually recognizes this foreign DNA or RNA part of the virus. It can go there and it can do cuts. What does cutting mean? It's just like DNA is cut, then it should be still there, right? No, DNA works like it just goes in one flow. Because when you're reading, just imagine you cut one part of the paragraph from the book, you just put it somewhere random, you don't know where to follow it. It's just really getting lost at this point. And if they can repair this system, then this information from the virus will be just done. And this is how CRISPR works. But why is it important? Why is it really, really important to have CRISPR, and why did it really open a new area? And actually, a couple of years ago, they won the Nobel Prize. The people who actually make this CRISPR work in a way that we can make really precise cutting. And this is the most important part. We can do really high efficiency and precise cutting. We know exactly which base pair is going to cut. We can control this. It's really easy, actually, when you know about this. You just have this targeting part that you need to put it into this protein. It just goes there, cut the cell, job's done. So, sounds easy. But the next thing that we're using, and it's going to be, might be a bit complicated, it is again CRISPR-based genome engineering tool, but it's a new one, let's say. Because what we're trying to do, as I talked about earlier, we try to understand how this disease works, right? How from the stem cells come into AML. So we need to use these mutations, we need to give these mutations to the cells, and then see how they're going to follow. But if we're doing the double cuts, strands to the cells, most of them will survive, but first of all, they don't like it, because these are sensitive cells that really divas, let's say, and they're just going to die, some of them. And then you also have some responses that you got from the DNA cutting, because they're going to have responses for this. And this is what you don't want. And in here, this base editor comes into the rescue, because it just does one cut. I'll explain later why. And then next thing will be, we will change C to T base. Normally, it doesn't mean anything, right? It's just like another base for you. But all of the stop codons, all of the parts that telling DNA to say, okay, stop, do not make any more protein, or like, do not continue this protein, is basically starting with the T base. So this is the most important part. If we can change the C to T in the right location, we can stop the production of certain protein. And protein means that, the DNA means that the protein production. So we will actually stop the protein production. Let's say you have a functional protein that works normally, and then you give the cuts, the stopping signal before that it finishes, and it's in the early phase, let's say, and then you're going to have this functional protein. And that's how you actually stop the cells producing this certain protein without giving them a major trauma, let's say. And this cutting is important because the cell wants to repair this, and when they see that the cut in this template, they want to use the other template to say, I need to repair this. And they actually use this newly made T base that we introduced as a normal base, so we actually trick the cells to change them into the normal part, and then you're going to have seamless, super nice T in the new area that you want. This is working quite nicely. And I'm going to explain how we do the project outline, like what is the things that we are actually doing. So as I said, we wanted to understand how stem cells are getting this phenotype, these mutations, and how it's going to proceed later on. So we're having cord blood, umbilical cord blood stem cells. These are basically coming from the placenta, umbilical cord blood, when woman gives birth, and they are okay for using these stem cells to use in the research. Otherwise, we cannot use it. And these should be really healthy, no mutation, super clean stem cells because they're coming from the babies, if you think about it. And then basically just giving this simple mutation whatever gene that you want to check, because you can give multiple, you just want to see one variable in the gene to see how this one's actually affecting the cells. And then we're going to have two different stem cells. Super simple. And then we're checking different stuff, how the editing of this works, how they're actually making colonies, because colonies, like all this hierarchy, if you remember, how does it look different, and then we need to put them in the living organism to see how it's working. Best would be to use humans, but as you can imagine, it's the parts we can use humans, so the mice is the next best reliable method that we can use. And I'm just going to talk about some of the information, results that we have, because I cannot get so much into detail, it's going to get more and more complex. The simplest one is when we put mutated stem cells among all of them. Let's say on the left you have healthy, on the right you have one mutated stem cells. In the next part, you're having normal production in the healthy cells, but in here the mutated stem cells are growing more. This is what we see actually. They're really growing way more than the normal cells. And after a while, they are overgrowing much more than healthy cells. Actually, in the mouse, that what I am seeing is if you put, let's say, 30% of this stem cells, in 16 weeks, you nearly have 98 to 100% of the mutated cells. So they're increasing so much that they are actually growing way more than the healthy cells. But it makes sense, right? Because we said that in AML, the cells need to overgrow so much that they don't give any space for the healthy stem cells. And it's actually happening. But these mice, they don't have any AML. They're having some other problems. They're having some blood loss or this cardiovascular problems, yes. But AML needs to take way longer. Because for AML to happen, you need to have way more myeloid cells. And this we don't see it yet, because you need to check the mice way longer. And you see in the healthy ones, we have the mouse and the mutated cells, they're not so happy, they have some other problems as well. And then you also see early dying out of this mutations that they have. And this is the problem that we're trying to really understand how we can actually check what is going on, because apparently mutation has an effect. But it is not AML that's so strikingly that we'll see myeloid cells. And this is really what we're trying to understand, what is changing in the mouse, what are the problems that we are having, not problems, but like what are the differences that we're having. And the ideal scenario at the end of my PhD, if we really make it properly, having some results, and if I'm really lucky, you can see some parts that will actually stop this growing of the mutated stem cells before they actually went into the myeloid leukemia. And this will be the ideal scenario, but as you can imagine, it's like really long shots. It's really, really hard to have. But this is just the outline of what we're having, and I'll keep you updated. So thank you for your attention. This was
questions?
What a nice talk. Thanks a lot. Awesome. Thanks that you started, very basic. So you mentioned these chip mutations, right? With these kind of pre-steps to getting super mutant. Can you think of a situation where it makes sense to do screening, to implement a screening program? And what parameter would you need to come up with something that is useful?
You mean screening in the patients when they come? And
Screening in the population, in patients, whatever screening program.
This is really nice idea, really nice question. And we also sometimes having this question as well. It will be already known which mutations that we need to check. As I said, we know what kind of mutations are there, and we know there's a correlation, because in order to have these mutations to be considered as CHIP, it stands for clonal indeterminate potential, because we know that we don't know what they're going to do. And we know the mutations that you can check on the patients, for example, you can find it, but if the patient is not there, a healthy person comes to the clinic, you need to tell them, yeah, we want to screen you for this kind of mutations. You might have it, but what will the doctors will tell the patients? Because just because you have these mutations doesn't mean that you're going to have AML or the problems. They might live really healthy life. die healthily. There's going to be no problem. But just because there's a chance of getting AML, a chance of getting cardiovascular diseases, you might put the fear in the people because they're going to think that, okay, I might die out of heart attack, I might die out of leukemia, and the problem is currently there's nothing you can do. Like you can say to the patient or this person that comes to the clinic, yeah, do enough exercise, go around, live healthily, do not eat fatty stuff, do not smoke more. I don't know, like this kind of stuff, which sounds super general and sounds really cheesy, let's say, but it's really helping actually in this case. And there is nothing else we can do. And you need to inform the patient what you screen for and what are the results. So the information that we gain is not helping the patient. That's the problem. If we have certain drugs that we can use for the patients, let's say for the TET2, if we have a certain drug that really stops the TET2 production, we can say that, yeah, we're suspecting that you're going to have this mutation, let us check. You might just check for these mutations that you have the drugs for, and if one of them is present, you can say that, yeah, you're going to start with this treatment. That might work out. That might actually will be ideal scenario, and this is the later stages that we're hoping to get. But before that, right now, having this information is not going to help neither us nor the patient, and that's the problem because we try to understand how they're going. And but good idea, yes, everyone wants to know, but I don't want to live in fear.
How about with the drug and then?
I'm trying my best. I'm going to go back to lab now.
Further questions?
I just want to add on this. But even for just like having a closer monitoring of those people, if you detect the mutation and you know that those mutations make the patient more likely to have that, then you have the information for you at least is useful right now because you're like, you know that you will need maybe a little bit more monitoring than the rest of the population. And so then we could have those people have a goal that, I don't know, every 2 years, 3 years, whatever, they need to do an extra exam. We have that for other diseases, like to have exams regularly to see how at certain age, and then depending on the results, you need to do other things. So I think this information could be for that.
Yes, true. But again, I'm coming to the part that these people are healthy. And you need to say that, yeah, we need to monitor you. And it's like, why?
Cancer or like those type of things are all about timing. And so if you know that they're more likely to have this,
yeah, but there's 99% of the chance that they might not get anything at all. And if you say to the person that, yeah, come in 3 years, we're going to monitor you, I mean, I guess you can tell. I'm not MD, so I don't know how it works. We can ask MD that. No, she said no.
So it's rather a question of money, actually, as well. So there is a ton of mutations like these, and you could always, check for cancer, but that's why we have this annual health checkup, which doesn't really find anything most of the time.
Well, tell us that.
I'm sorry. Yeah, no, but you only, so the breast cancer, the BRCA gene, that's one of the genes where you actually know the chance is so high that it's worth doing a very often check-up. But in most of the genes, it's so rare that you don't do it because,
Yeah, that's fair.
I can just how far is CRISPR-Cas9 actually used in people? Is there any way that is on the way being used for any disease, not just cancer?
Actually, this was one question that I did want the moderators to ask. If there's a normal question, I can just answer. It was really nice because there are clinical trials actually with CRISPR-Cas9 that are used for. Okay, for CRISPR-Cas9, what I know is with the CCR5 for the key helper receptor that found in the 8 patients, they can knock this one out. So the virus, the HIV virus, cannot enter to the cells. So even though you have the virus, you actually don't suffer from AIDS. So this is really, it depends, irregardless of the virus load that you have. So you're not going to have an AIDS. This is one thing that they're trying, it's in the clinical trials. And one thing that I found really cool with the beta editor, they are actually having some clinical trials with cystic fibrosis. They are actually, because it's coming from one mutation, and there are different kinds of beta editors as well, it's not just this. They are now expanding more, and they try to figure out how we can do more changes. And one thing is actually changing this mutation back from the stem cells to the patient, and then give back to the patient and see if it's going to work out. All of them are in trials. Sounds nice. because it's actually not, I mean, it's going to be a bit subjective, but it's not that hard when you know what to do. It's just a matter of like, how this is going to progress, how the body of the humans will react to it. Because if I have stem cells that are coming from people, and I can introduce mutations, right? If I know where to target, I can change these mutations as well. Just it's going to be less than probably 1,000 euros. It's not that hard actually to have this. The problem is that there are a lot of safety regulations. You cannot just change something and give back to humans, because there might be some unforeseen effects that you can have. That's why we have clinical trials. That's why there's a lot of checkups are like happening. But true, yes, they are actually using these to target different parts of the diseases that they can target. Some are easy, like cystic fibrosis is really lucky because it's one mutation and they can target. Or CCR5, they know it's going to help, it's also lucky. But for some diseases, it's not so easy because the recognition is not so simple. You need to have certain type of basis in this forecast to go and recognize this. If you don't have this, you cannot change it. And that's the problem, that not every gene can be correctable. It's not like a magic wand that you just put it there, it goes and saves you. It doesn't work like that. That's the problem. There are clinical studies, so they're actually using it. So.
How many bases can CRISPR-Cas recognize and cut out and exchange?
So, just by itself, CRISPR-Cas just goes and cuts. That's it. It doesn't do anything else. But we have some different like versions. There's one of them called like because the cells want to repair the DNA, right? If you cut the DNA in the middle, they're gonna say, okay, I need to repair it, and I need to find something, like some information to repair. You can give them some DNA strands. Just some parts are recognized on the both cut sites. These are like homology arms. And then you put whatever gene that you want in here, and the cells will say, this is the same template. I'm gonna read this, and this whole part is actually integrated into the genome. This happens, we're doing a lot. I have a colleague that actually, colleagues in here that are also doing the same thing. We are actually giving the parts into the cells, and the cells will just have these proteins produced with no problem. Actually, one of the things that my colleague was trying to have is, you have a certain type of mutation in a certain gene, and what you try to do is cut the gene before this mutation and give the healthy information to the rest of the protein to produce the healthy protein. It is easier said than done, so it's really a lot of work, and there are a lot of unforeseen things might happen. But these are stuff that we are eligible to do it. These are stuff that you can delete some part of the DNA, you can introduce.
How long can be the cards? How many days?
For a health integration, it's going to be 4.7 kg.
Yeah.
4.5, so it's at 4,700. So it's not so big, but it is helping to put something in there. The rest of the protein, let's say, you can have it. You can even put some certain type of fluorescent proteins to check which cells actually got these parts. So they will be, we can make them like green, red, blue, whatever. And then when you see the color, you say that, our gene is integrated there. So you know which ones are actually having these mutations. And this is actually one thing that we are doing in the lab. So we are doing one healthy, one sick alleles of the genome, and just putting it there with two different colors. So you know that one should be wild type, one should be green. So you have different genes integration. So these kind of stuff you can do, but it's getting more and more technical, and it's just, I think I know that the one clinical trial that we're using HDR template like this, they're putting the healthy part of the cell into the protein before it has mutations, but they had some unforeseen results happen, and some of the patients died, and they need to stop the production of the part. So it's not as easy as it sounds. It's not super simple, and these kind of parts that need to be really considered. It's not so easy, let's say, but it is there. Every day is just getting something new. Every day is like new regulations or make it more precise are happening. So it's a good time to live, let's say. That, it will be nice to have, I don't know, in 50 years maybe you'll have most of the genes that you can change easily, you don't know. Because if you think about it, the time of CRISPR-Cas9 has been found, not found actually, it's found in the 80s, 90s, but actually can be used to actually have this precision genome editing was 2012, I guess. It was 12 years ago, and after this, the studies are skyrocketed. So now everyone knows about it. Like, also here, you heard like the most of audience. I don't know, maybe all of you are studying the Medellin, I have no idea. That would be really unlucky. But then you have the CRISPR, and everyone knows about it. And this is the part that you can do a lot, and it's just improving over time.
What do you think about biohackers? I don't know if you heard about it. That the general public can buy CRISPR kits and people at home trying to inject themselves with bigger muscles.
Yeah. It's not going to work. I know. I watched what was the series? I forgot the name, but the guy is basically just putting the plastic.
It's called biohackers.
It's biohackers? Okay. Yeah. It's not going to work because they're basically just getting plasmids. Plasmids are really big pieces of DNA, this round DNA. And in order to put them into the cells, we need to treat the cells in a certain way that they will just get integrated with electroporation or maybe some oil droplets and whatnot. And the efficiency is not good. Cells do not like it so much. And he's just injecting himself with this. You have all these myeloid cells that I mentioned about. They recognize important parts. He's going to have all these myeloid cells that will come there, neutrophils, macrophages, whatever. They recognize there's something foreign. They're going to eat it. That's it. They're not going to get into the cells even. So it's not, and he needs to do millions or billions of injections for him, for all his muscles to be grown. I mean, the idea is there, I know, but it's just like super hard. And I think it's a good thing that they cannot be able to do it. But also, as I said, it needs to be really controlled. If you just really get it out, you don't know what might happen. You cannot control it. And this is something that really needs to.
Thank you very much for your great presentation. Round of applause. Great time. It was a really lively discussion, and I'm really happy with this crowd. Everything was nice. And here is a small present for you from us. And I would also like to give a present to someone in the audience. So now I remember that there was only one person that answered one question of yours. I think it was you. Was it? Yeah, I think so, but the number was...
the 100 billion.
So, you get a glass. Here you go. And we wanted to ask an additional question. Because we also have a bag. So, please, moderators, look at the people. What is this animal called on my shirt? Who was first? Her.
So you get our bag. Yeah, I'm the audience side.
So,